Hello and welcome to Nikolai's genetics lessons, and the first question of the day: how many different alleles can be detected per heterozygous individual with a specific single locus probe in DNA fingerprint analysis? And here's a variance of the answers. You would be able to answer this question if you'll take a look at this picture. What we see here 23 chromosomes, actually 24 chromosomes, because both sex chromosomes are present here, but the rest are present in one copy, and sex chromosomes are present here in two copies. So this is male karyotype. And what we see here different loci, and at each locus we can get a certain number of repeats of the core sequence. For example, let's analyze the first locus. This person, for this particular locus, which is D8, so on the chromosome number 8, in this locus has two repeats. On one chromosome that he inherits from the mother side, he has 14 repeats of the core sequence, and on the chromosome inherited from the father side also has 14 repeats of the core sequence. And in this column we see how many repeats we expect. Alleles can be between 9 and 17 repeats of the core sequence. Here between 6 and 14, 6 and 15, 12 and 19. So you know that there are 7 alleles present in a gene pool, and each allele would be with different frequencies. Sometimes people can be homozygous, sometimes can be heterozygous. But as you understand, because we have only 2 chromosomes for each pair, a person who is heterozygous for certain locus only can have 2 different alleles. One on the chromosome inherited from the father side, and one on the chromosome inherited from the mother side. So the correct answer is going to be answer B. No more than 2 alleles per one locus. Those in a gene pool, we may have more than 2 alleles present. For example, 7 alleles can be present for one locus in a gene pool, but not more than 2 different alleles we may have per one locus. What we see here we call genetic profile or genetic fingerprint, and it is easier to win $1 billion in lottery than randomly predict genotype genetic fingerprint of the person. Now we can move to the second question. Which of the following is not a potential source of DNA for forensic analysis? Not much to explain here, you just have to memorize that only red blood cells doesn't have nucleus, and all the rest cells have nucleus and has DNA, has chromosomes, and we can perform genetic analysis and make genetic fingerprint. But red blood cells early in the development stage would be denucleated and would be released in the blood without nucleus. Red blood cells do not live longer than 3-4 months and would be constantly replenished. And also you may wonder then how we can use blood in order to do genetic analysis if red blood cells doesn't have nucleus, except red blood cells, which make about 45% of the blood. There are also white blood cells which only make couple percent, but this white blood cells has nucleus, so we use white blood cells in order to do genetic analysis and profiling of the person. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.